So, in a couple of sessions now, I'm going to discuss with you a very important aspect, which is associated with biotechnology and also with our understanding of biological systems and the organisms. And I'm referring to the variation in living forms which exist in nature. So we are going to talk about what is the genetic basis of this variation. And also, as we know that this natural variation is transmitted from one generation to another, you know. And this aspect was systematically uh, looked at by Mendel in 1860 60s or so, and he came up with certain principles which are which explain that how the various characteristics or what he called as traits are transmitted from one generation to another. Uh, but then I'm not going to take you to what Mendel's did, but I'm going to take some leads from Mendel's work and then explain it in today's terms, the genetic basis of which exists in living world. You know, for example, the question that what controls this natural variation, it was now we understand that uh, the it is the allelic difference which exists at the gene level which controls a specific trait. You know? So what I'm trying to say is it is the allelic difference at gene level which controls a specific trait. So in this, let us define what is gene and what is allele. You know, so gene basically is a piece of DNA, you know, which controls the expression of a trait. So gene is a piece of DNA which controls the expression of a trait. So gene and trait, you know, and then what is allele? So allele is basically the alternative form of a gene. It means a gene exists in more than one form, or at least in two forms. So it has it has two alternatives. So he can have one kind of trait, and if there is another form present, it will have another kinds of trait. Okay, so that's how the the natural variation existing in the nature could be explained. You know, so then if we look at or want to understand it more clearly, the question that what is the difference between genes and alleles for Mendel's trait? You know, so there is a classical picture what Mendel's took as traits like plant height, you know, and the seed coat shape or seed shape. So he considered the two traits or features of a plant. He worked with pea plants. So he observed that certain plants were tall and other plants were short, or he called them as dwarf. Okay. So similarly, when he looked at the shape of the seed, some were smooth seed coat surface and others were having wrinkled surface. Okay. So when we see the relationship, the plant height is expressed tall and short. So these are the alleles. These are two different forms of a trait. So tallness and dwarfness or shortness are the two alternative forms of a trait, which are controlled by different genes. Similarly here, the smooth surface and the wrinkled surface are the two alleles of the same trait. Trait is the seed coat quality. So some seeds have smooth surface, some has wrinkled surface. So these are the alternative traits, or what we call alleles. Okay, so the trait is the seed shape, and alleles are the smoothness and the wrinkledness. So alternative forms of the alleles or the genes. So this was observed by Mendel, and then it was later on further explained at the gene level. So it means that there is a genetic continuum. What is continuum I'm talking about? That there is a relationship between gene, alleles, and phenotype. Genes, alleles, and phenotype. There is a relationship. Okay. So when we talk of alleles, so alleles, we have to understand when we say alternative alleles. Okay. So an allele must be similar. If we have to define that, the allele must be similar enough to control the same trait, but different enough to create different phenotypes. Okay, so allele must be similar enough to control the same trait, and different enough to create different phenotypes. Okay, so from gene to allele, and then the phenotype. Phenotype is the different forms of the trait. Like in the previous example, I showed the height, tallness, and the dwarfness, the seed quality of the seed coat, the smooth and the wrinkled. So these are the alleles which are similar enough to control the same trait in terms of height and the shape of the seed coat, and different enough to give different phenotype. So they are controlling the same phenotype, but different enough to give uh, different phenotypes. They are controlling the same trait, but different enough to give different phenotypes. Okay. So further, this was the gene basis of this was also understood, that how allelic differences of Mendel's gene controls the plant height. So, I mean, subsequently, when the functional genomics came into existence, then these studies were redone. And now we know that there is a gene which is responsible for gibberellic acid synthesis, and it is the gibberellin or plant hormone which is responsible for cell division and elongation, and in other words, it controls the height. So what we're seeing here, that this gene, that is gibberellin 3 beta hydroxylase, this gene is responsible for the final synthesis of gibberellic acid, or GA1, which regulates the cell elongation and cell division. And if there is a mutation in this gene, and very specifically, if a alanine amino acid is converted to threonine in the final protein, then this enzyme loses its function, and you have a mutant protein generated, which is 1 by 20th time as less active as the original one, that is the wild type. Okay, so single mutation in one amino acid can give you an alternative allele, which brings about shortening of the plant. So the dwarfism is created at the gene level by this mutation in the normal gene. So this gene controls the height of the gene. If it is intact, then you have the long plant, and gets mutated, point mutation, then you see the dwarf plants are generated. Okay, so from gene to allele to phenotype. So now we know what controls the dolphin. Similarly, the other trait which Mendel considered, the quality of the seed coats. Now that is, now we know is controlled by another enzyme called starch branching enzyme, SBE, isoform 1. Now this, and it is coded by a gene. Okay. Now, a function of this gene is to add branches to the starch, you know, so to generate complex carbohydrates in the at the seed level. Now, if there is a mutation by transposon insertion into this gene, so effect of this mutation in this gene is that at the maturation, seed loses more water and it creates wrinkles. Okay, so again, this smoothness is provided by the intact gene and its ability to synthesize complex carbohydrates. But a mutation in this is responsible for seed to lose more water and create wrinkles in them. Okay, so uh, so uh, so to summarize what I had been talking about is the concept which came up based on these kind of studies that if a trait in either plant height or this is controlled by gene at the DNA level, which transcribes to RNA and then translates to protein, and then protein reflects to the phenotype. That is either the plant height or the seed coat. This is an example, but now we know that that's how the all the traits are controlled from uh, DNA, from gene to protein, and protein to phenotype. So the simple relationship has been generated or generated between the gene and the phenotype via proteins. Okay, and a change in this gene by way of mutation, gives you the alternative phenotype, which in case Mendel's observed without this knowledge, and he just says that there are allelic differences he considered at that point of time without knowing anything what I'm talking about in terms of genes, proteins, RNA, DNA, etc. Okay, so he only talked about alternative traits being present. So height, for example, he considered uh, as existing in two alternative forms, tallness and dwarfness, that is capital T, small t, and all that. I'm not going into the details of that. So only point I'm trying to make is that today we understand that the genetic variation which is observed is controlled by this central dogma principles that any trait has a genetic basis or a gene for it, makes a protein, and then that protein controls the trait. Okay, now if the genes are responsible for the genetic variation and all the traits which an individual organism has, then how coordinatedly these genes take care of this variation without losing their identity? And it is these kind of questions when people looked at uh, in 60s of last century, uh, first in the bacterial systems, that how these genes are regulated or controlled, whether there is, I mean, how these genes, for example, sitting here, makes protein, and these proteins get associated with the phenotype. So how these genes are expressing themselves. So another terminology came into existence, that is the gene expression. So how these genes are expressing themselves. This question was addressed both in prokaryotes and later on the eukaryotic systems were also exploited, and we now understand that how these genes they are regulated, their expression is controlled and regulated in prokaryotes and in eukaryotes, in animals, in plants, in humans. So we have clear-cut understanding about this aspect, and I'm going to very briefly touch upon this. So let us look at in the what happens in say how this gene regulation takes place, or expression, gene expression is regulated. So what we understand today is that all living organisms in general, starting from prokaryotes to eukaryotes, they have two class of genes. It means their genome, in genome also, it is the active genome part which has the genes. It has two types of genes, and that is based on their expression. This classification has been done. So there is a class of gene which expresses themselves all the time in all the cells of an organism. Okay, and we call them as constitutively expressing genes, or the housekeeping genes. Basically, the role of these housekeeping genes is to keep the cell in living condition. I mean, if they don't express, the cell dies. So housekeeping genes are responsible to keep the cell alive. Okay. So there is a part of genome which expresses, again, I'm repeating myself to make you understand that there are two types of gene complexes or groups. One which expresses all the time in an organism, in a cell, and this group of genes we call housekeeping genes or constitutively expressing genes. Okay, and they are responsible for keeping the cell alive. And the another class of or group of genes, and these genes are simple. They have a simple structure actually. Okay, a simple structure. What I mean is a promoter and this and the structural gene and the terminator. Okay, so there is no other part in the gene. So they have a simple structure. Whereas another group, what we call controllable genes. Now there is another group of genes which are controlled. They do not express in all the cells all the time. Their expression is guided and controlled by cell itself or by external factors. So until unless those factors are active, this group of genes will not express themselves. So they are highly controllable class of genes. Okay. It means their expression takes place only when cell needs it. Okay. Otherwise, they remain dormant or silent. So they don't express themselves. Okay. So they are, I mean, either when I talk about the regulated expression, in a cell, you have four kinds of gene expression activities. Either a gene will start overexpressing itself, means the product will be more than the basal level activity, or it will start I mean the expression goes down, okay, so that the product is made less. And the third is a gene which is not expressing at all in a cell starts expressing, or a gene which was expressing, it stops expressing. So there are four kinds of activities you have in the controllable group of genes, that either a gene will start expressing, which is not expressing in the cell, or it stops expressing, or it is overproduced, overexpressed or underexpressed. So, and these all four kinds of expression control are highly regulated, and we have a clear-cut understanding about most of these phenomena that happen. Now, if we look at the gene structure is uh, more complicated as compared to the housekeeping genes, where I told the structure is simple. Here it is more complicated in the form of that they have some response elements, they have activators, repressors, etc., which act upon the transcription unit and brings about these changes which I was referring to. So we will look at first in the prokaryotic system that how this expression is regulated very quickly. I know you have gone through. So let us look at it. So there are two ways in which a prokaryotic cell, bacterial cell, controls the expression of genes. One is promoter recognition, and second one is the transcription elongation, or what we call attenuation. So there are two ways by which a prokaryotic gene's expression is regulated. Now, the difference between prokaryotic and eukaryotic transcription unit is that uh, the structural or operon part, what we call, is in all the genes which are responsible for a pathway, they are located side by side in the same operon. In other words, what I'm trying to say is that all the genes which are required for a metabolic process or pathway uh, are expressed, either all of them are expressed or none of them is expressed. Okay, so it is all or none kind of thing. Whereas in eukaryotes, all individual genes involved in a metabolic pathway are located and expressed individually, not collectively. So in prokaryotes, you have this kind of operon system, which you can say it's a complex, in the sense that all the genes involved in a pathway are co-expressed, or their expression is blocked. So this is also you have to keep in mind. So we have two major categories of regulatory systems in prokaryotes. One by way of recognizing promoter, and another one by way of attenuation. And also, I should tell you that in a cell, you have two types of metabolic processes, that is the catabolic processes and anabolic processes. It means the synthetic processes and the degradable processes. And both these kinds of processes are regulated differently, So, the gene expression is divided based on the kind of process they are controlling or regulating. So, if we look at some of the definitions which have come from studies based on the prokaryotes, and that was the first system to be studied. And let me tell you that whatever was given to in 1960s with respect to prokaryotes, it holds true for eukaryotes, and we use the same terminology. Nothing has been changed. Okay. So in a operon, as I mentioned, a few genes that are controlled collectively by one promoter. And that's what I was trying to tell you, is that all the genes involved in the metabolic process are controlled by a single promoter. So operon system has that few structural genes are co-expressed by one promoter. And then and they are controlled by cis-acting promoter and operator parts. Okay, we will see that what is promoter and operator part. They sit close to the structural gene part to regulate the expression, and it's regulation by transacting repressors or the activators. I mean, typically, if you see uh, a operon structure is something like this. Okay, you have a structural gene. This is from here to here. You have the operon. So you have the structural genes, for example, and then this is the control region, which controls the expression of these genes, and it has a promoter and it has an operator. You know, and then you have regulatory genes, which may produce a repressor or an activator. They can be present anywhere in the genome or in the closer proximity of the operon, or it may be at a distance. Because what is required here is the gene product, which can diffuse and approach their site of action. So a operon is very simply explained like this. You have two components, structural genes and the control region. Control region has a promoter and a operator, and then the regulatory gene, which produces a repressor or a activator. Okay, now very quickly, if you remember what happens in the Lac OPERON, which was the first system to be understood, because it is, I mean, it was seen that lactose is a substrate which bacteria uses as a carbon source. So it is taken up inside and it gets degraded into glucose, which is the simpler form and which cell requires. So how this metabolism takes place, you know, how this operon is regulated, you know. So what happens when you have, in general, okay, okay, let we go to this part. So what happens when a bacteria is grown in presence of lactose? The lactose is taken inside the cell, and like here. So if you when the lactose is goes inside, you know, the RNA polymerase binds to this operator. Um, um, and, um, um, Now, in presence of lactose, okay, here in presence of lactose, this is what happens, that um, that RNA polymerase binds to this and brings about the transcription of these LAGZY18 coding for three proteins which are responsible for lactose uptake and then the degradation into glucose. Okay, so that is in presence of lactose when the bacteria is growing. Okay, now but when lactose is not there, then what happens? Then the expression of these genes is blocked. And how that blockage takes place? That is here. That is through this inhibitor. Now, this inhibitor, you know, this inhibitor goes and binds to operator in absence of lactose. And RNA polymerase, though bind here, but it is not allowed to transcribe. But when lactose is given, the lactose interacts with this inhibitor and brings about a 3D change in their conformation and doesn't allow to go and bind here, and the transcription goes on. Simple control mechanism of the Lac OPERON system, you know. So in presence of lactose, you see the expression of these genes. In absence of lactose, you don't see the expression of these genes because the products are not required. There is no lactose to be taken inside and degraded into simpler forms. So that is because of this inhibitor. This keeps this operon under check in absence of lactose. As soon as the lactose comes, it interacts with this inhibitor and doesn't allow it to go here to the operator region and brings about transcription. So simple Lac OPERON system was given. This is first system to be understood, and very clearly explaining what goes on and how these genes are regulated. I mean, it's again a scenario which was worked upon that if a cell is exposed to both lactose and glucose, then what was seen that though the lactose is there, but Lac OPERON doesn't work until the glucose is completely exhausted. So the presence of glucose keep this Lac OPERON under check. Now that was also looked at from that what is controlling this, that why even in presence of lactose, the Lac OPERON is not activated, and that is because of the involvement of glucose metabolism and that the involvement of the cyclic AMP and cyclic AMP receptor protein. Now, this is the complex, which is is required for tightly binding the RNA polymerase to Lac OPERON. So in so long, and if in presence of glucose, this protein is not made in sufficient amounts. So the RNA polymerase binding to Lac OPERON promoter is weak, and it doesn't allow transcription to take place. But once the glucose is exhausted, this cyclic AMP CRP production is increased, and this complex then binds to the operator part, the promoter part, and allows a stronger binding of RNA polymerase to the promoter and brings about the transcription. So this OPERON actually is under both negative and positive control. Okay. Now there is another scenario wherein the OPERON is controlled by the product. So long that product is there, the OPERON is kept in check. And once the product is not there, the OPERON start expressing. Simple example is the tryptophan amino acid biosynthetic OPERON, or Trp OPERON, what we call it. It is an example of repressible OPERON. So what happens here? So long there is tryptophan available in the cell, this is kept under check. This doesn't express. Once the concentration of tryptophan goes down and cell needs the tryptophan to be made, then this OPERON starts expressing. So how this is explained, so in absence of tryp, when there is no tryptophan in the cell, the RNA polymerase goes and binds to the tryp promoter and brings about the expression. Okay, and there is another, a repressor which is present, and it is not binding to the or interacting with the polymerase, So, and that is in absence of tryptophan. The polymerase goes and binds it. But then, when this is in absence of tryptophan, but when the tryptophan is present, how this is kept in check is that, see, this is the repressor, it interacts with tryptophan, and this complex or this interaction brings about a conformational change in the receptor protein, which goes and binds to this Trp operator and doesn't allow polymerase to bind here, and the OPERON is kept under check. Okay. Now, if you then there was another question associated with this, was that the cell may have tryptophan, but it may not be sufficient to bring about the cellular functions of protein synthesis. So it means the cell also senses that fine concentration of tryptophan. Until unless it goes down to that level, the Trp OPERON doesn't start synthesizing or becoming functional. So what we call a fine-tuned control is exerted in this OPERON. So it is all or none is fine, that so long there is enough tryptophan or there is no tryptophan, and the Trp OPERON works, and in presence of tryptophan, it is kept under check. But the question I'm raising here is that there is tryptophan, but it is not good enough. It is not in good enough concentrations to bring about the cellular functions like protein synthesis. This is involved, tryptophan is amino acid involved in protein synthesis. Okay, so that kind of fine control is basically controlled by attenuation mechanism. That is an example where translation controls the transcription in prokaryotes very, very effectively, and that is through a region, what we call, which sits in between operator within the promoter region, we call leader region. And leader region has the 28 amino acids, uh, it codes for a protein which we call leader protein, which is 28 amino acid, and these sequences are present in all sorts of region frames, and it has a stop codon also, So this is very, very important, and this fine control which I was referring to is exerted by this region, actually. Okay, and what is this? How it happens? So when the transcription is going on, the RNA polymerase moves forward, and the translation also, as you know, in prokaryote, transcription translations are coupled, So when this is moving on, so you have this region which is capable of forming the pairing. These are the regions which can one, two, and three, and four actually. So what happens when there is cell is starving and there is no tryptophan, then the RNA polymerase is transcribing, and you have, this is the one region, sorry, 1, 2, 3, and 4. These are the regions which are capable of forming pairing. I mean, this is the RNA region, mRNA region basically. So RNA polymerase, when there is no tryptophan, or the cell is starving of tryptophan, the translating system machinery stops here because there is no tryptophan to be added here. And this kind of pairing takes place between the mRNA transcribed by RNA polymerase, So 2 and 3, So the transcription continues, and you see but then when there is some amount of transcription is present, or transcription is present, then what happens? You have this 2 and 3 pairing and bringing about this kind of structure wherein and creating the stop stem loop structure, which is the transcription stops, which gives the transcription stop signal basically, and the transcription is stopped here. So this is the region actually, what we call attenuator, or the process we call attenuation, where translation guides the transcription process. So these are the kind of regulatory mechanisms which have been worked out, studied for prokaryotic system, and it explains almost all pathways, catabolic and anabolic pathways, which go on in a bacterial cell. Okay, so now after this, the question was to look at what is going on in the eukaryotic system. Okay, so with this kind of understanding and background, the further work went on to understand how a eukaryotic gene expresses itself and how their expression is regulated, and I will look at that in my next session.